you for joining me today at Oncomine World. I'm happy to present today on Oncomine Comprehensive Genomic Profiling with OCA+. Let me begin with some background on the value of comprehensive genomic profiling, or CGP. Simply put, CGP is defined as the ability to simultaneously detect all necessary variants in a single test, which gives us the value of providing maximum insight into each sample, including its oncogenic drivers. Some complex biomarkers, such as genomic instability and TMB, really require a large genomic footprint, and therefore a large panel of more than 500 genes spanning multiple biomarkers is one way to implement comprehensive genomic profiling. CGP brings a lot of promise to clinical research, but many offerings have limitations or challenges that really prevent more broad implementation. These limitations including being too limited, of having poor sample success. Many CGP solutions can require a large amount of DNA, such as 20 nanograms. And with FFPE samples, tissue amounts are finite, and that can be a challenge. In addition, some technologies do not work well with poor quality samples. Another limitation is being too slow or having very long turnaround times. The library preparation for some techniques, such as hybrid capture, can take several days just with a single step. This adds considerable time to the overall turnaround time to go from sample to results. A third limitation would be being too complex to implement. This includes the lack of an end-to-end solution as well as a full bioinformatics pipeline. Having to piecemeal different solutions mean that these individual investments can add up. There's also extra work to ensure compatibility, whether it's software or reagents. Without an end-to-end workflow, this could be rather complex work to implement. In addition, another limitation is being too incomplete by not being able to analyze all of the relevant biomarkers. The exploding research means there's rapid growth, and being able to make sure you can detect all biomarkers is super critical. One example would be HRD as a biomarker, which has been getting a lot of attention of late. All of these limitations, we feel, has really hindered the broad implementation of comprehensive genomic profiling. With this, we want to offer Oncomine Comprehensive Plus on our Genexus platform to really be able to enable next-day CGP without compromise. We do this by providing a solution of a very large gene, looking across 500 genes, looking at all of the relevant biomarkers in both DNA and RNA, covering single gene biomarkers such as SNVs, indels, fusions, and copy number variants, as well as BRCA1 and BRCA2 large genomic alterations. But really expanding into the multiple gene biomarkers, such as the complex biomarkers of genomic instability, tumor mutational burden, and MSI. We're going to go into more details of our genomic instability metric later. And OCA Plus offers this all using our tried and true AmpliSeq technology, which utilizes low DNA input and offers high testing success. We also include a full end-to-end workflow, including automation as well as bioinformatics, to give an ease of use to be able to not limit implementation of comprehensive genomic profiling. With OCA Plus on Genexus, we really think we can enable next-day CGP results without compromise. A little bit more into testing success rate as well as turnaround time, two factors that are really key to clinical laboratories. On this slide, we kind of highlight two publications by our customers to speak to these 2 points. First is with Strata Oncology. Strata Oncology is a US-based laboratory where they were able to demonstrate using a 400-gene AmpliSeq panel across a very large clinical cohort of greater than 30,000 samples to really have good testing success of 94.2%. This study was done on our Ion Gene Studio platform, and they were able to get a relatively good turnaround time of approximately 7 days. Moving on to the next study, this is with our SCRUM study in Japan. For those that are not familiar, SCRUM is Japan's first industry academic collaborative cancer genome screening project for individualized medicine. This study, demonstrated with our 50-gene panel using our Genexus platform to be able to really improve on turnaround time, moving from over a week to now just 4 days within the Genexus system. We do understand both success rate as well as turnaround time can make a huge difference in clinical laboratory, And with OCA Plus, we take advantage of both by using AmpliSeq technology and now available soon on the Genexus platform. Our Genexus platform really will simplify comprehensive genomic profiling by giving a true end-to-end solution, going from sample all the way through report, using just 2 instruments and a single software with 20 minutes of hands-on time. 10 minutes of hands-on time on the Genexus purification instrument, and another 10 minutes on our Genexus sequencing instrument. All of this we feel would allow our customers to really implement comprehensive genomic profiling without compromise. In terms of the other limitation in regards to complex biomarkers, we really do hit all relevant biomarkers with OCA Plus. When we first launched OCA Plus on our Gene Studio platform, we offered TMB and MSI, and with a recent software update, we now also were able to unlock homologous recombination deficiency by providing our GIM. We're going to go into that in more detail. So more into homologous recombination deficiency as a biomarker. Let's give some very brief background in terms of what is HRD. Simply put, HRD is our inability to repair double-stranded DNA. In cells that are deficient in homologous recombination repair, which is a cellular pathway, cells will accumulate on genomic damage called genomic scarring, and this leads to genomic instability. In cells that are deficient in homologous recombination repair, when you also go in and hinder single-stranded break repair, for example, through PARP inhibition, this leads to cell death and a concept called synthetic lethality. Because of this, HRD is an emerging biomarker that really is a way to look into sensitivity to PARP inhibition. The background on HRD, there are different ways that you can measure HRD. This would be on both causes as well as consequences. And OCA Plus can do both. We have a very comprehensive approach by interrogating 46 different homologous recombination repair pathway genes, looking at different endpoints. We look into SNVs and indels and even copy number variants, as well as expanding onto gene-level loss of heterozygosity. And for BRCA1 and BRCA2, we also can look into large genomic alterations. In regards to genomic scarring, this is kind of the exciting new feature that we recently developed as a bioinformatic endpoint. Our genomic instability metric is our way to measure the consequence of HRD. The genomic instability metric, simply put, is a proprietary algorithm in our Genexus and IM Reporter software that looks into genomic scarring associated with homologous recombination deficiency. GIM is a summary of unbalanced copy number changes in autosomes using genomic segmentation. This is a quantitative metric from 0 to 100, and the higher the value, the more genomic instability is in the sample. In the next slide, we're going to go over some of the technical performance of our GIM. The first table really looks into genomic instability in cell lines, both positive and negative cell lines. In cell lines with known BRCA mutations, our GIM is able to provide a positive readout. And in the negative samples, such as NA12878, we're able to demonstrate that our GIM is negative in that cell line. In the middle plot, moving away from cell lines and looking into clinical samples, we're able to show that our GIM is able to stratify ovarian cancer samples. In a cohort of ovarian FFPE samples separated by BRCA mutations, on the left-hand side would be a benign or wild-type BRCA status, and on the right-hand in the orange bar is pathogenic BRCA variants, or otherwise known as HRD positive samples. And you can see our GIM is able to properly stratify between these two different samples. We get great performance moving over to the right between both Genexus and Gene Studio to show, very good concordance. And with that, this is kind of just more technical performance of GIM. I think we're more interested in knowing more about how our customers are using this. So we'll move on to the next talk and really be able to give you more details on how GIM is performing, with our next customer presentation. Thank you for your time, and I'm also available for Q&A during that session. Thank you.
Hello, I am Annette Staebler from Tuebingen University, Institute of Pathology. Today I'm going to present our results on a study on genomic instability metric from Oncomine Comprehensive Assay Plus. We did these studies together with our co-workers from the Department of Molecular Pathology, from the lab from Irena Bonsheim and Francisco Otto, and with support of our Chief of Service, Falco Fendt, also. This is a disclaimer from Thermo Fisher. I'm going to start with some background on homologous recombination deficiency as a therapeutic target. Homologous recombination deficiency leads to inability to repair double-strand breaks in the cells. So if there is homologous recombination deficiency, which I will from now on call HRD, the cells tend to accumulate genomic instability and genomic mistakes. This is how the inhibitors of PARP works. Then in that system, if you give a PARP inhibitor, the cells are not able to repair single-strand breaks. Those are converted into double-strand breaks, and then again, the inability to repair the double-strand breaks by homologous recombination takes in, and these cells accumulate even more genetic defects. Alternatively, they can use error-prone mechanisms as depicted down here in the right segment of the graph. And overall, if these cells are treated with PARP inhibitors, they will accumulate a lot of genetic instability, and eventually many of them will go to cell death. This is the reason why BRCA mutated tumors respond better to PARP inhibitors than tumors without mutations. This was one of the first studies looking at PARP inhibitors in ovarian cancer, leading to some of the most spectacular extension of progression-free survival in these patients, and mainly in BRCA mutated tumors. But there was also an increase in survival in tumors without BRCA mutation. So there had to be other mechanisms, HRD induced by defects of other genes, for example, which are not covered by BRCA mutations. Other studies have looked at HRD and BRCA mutation as predictive markers for the treatment with PARP inhibitors, and they've shown that even tumors without BRCA mutations were responding if you were able to demonstrate HRD in the tumors. This is here the PRIMA study that looked at niraparib. But in addition, HR proficient tumors would also respond to a lesser extent, but there was some response. So we need to think what happens in these tumors. Maybe the currently used assays are not completely covering all the cases, or there's yet another mechanism. In another study, PARP inhibitors were used in combination with another treatment like bevacizumab. This is like in the Paola study. Here in this study, a clear benefit was only seen in the cases that had HRD or cases with BRCA mutations. So if there is another baseline treatment given, apparently you really need to demonstrate sufficient HRD to have the full benefit of the PARP inhibitor treatment. So here are two good reasons why to look at other assays to look for HRD. One is we try to cover the cases that are maybe not detected by the currently available assays. And secondly, we want to be as precise as possible to assign patients to treatments where we know they are only going to respond or have the full benefit if we can demonstrate HRD or mutation in one of the BRCA genes or any of the other HRR genes that are involved in that same pathway. So how can we measure HRD? Well, we have basically two major ways to do this. One is we look at the causes of HRD, which is inactivation of genes in the HRR pathway, like, for example, BRCA1 or BRCA2, or some of the other components that are here depicted in this complicated network of different proteins involved in this homologous recombination. The other way is we're looking at the consequences. We look at the genomic scarring. We look at the different alterations that we find in the DNA, and these are some listed here. We will go on this in more detail later on. So this we can do by analyzing this tumor DNA in specific ways with the specific algorithms. We have also a third way, which is also mentioned in these ESMO recommendations. So one here, you have the detection of mutation in or silencing of the HRR genes. Then you have the genomic scars. But in addition, you could also look for functional characterization and look for functional HR deficiency if you study live tumor cells. This is a very sensitive method, but of course this is not always available and technically very cumbersome, you want to say. But there are people who prefer this, centers that prefer this assay, definitely. In order to look at the genomic scars, we have different targets that we can look at. They are sort of as an overview here, again, from this guideline paper. One is copy number variation, and looking at different scores, different types of copy number variations like LOH, NIA, or LST, which is also used in the GIS score. And then you can also use signatures that combine different features that take also into account single base substitution signatures, like, for example, HRD detect. Then also we have complicated algorithms that take into account multifactorial dimensions, also with some other tools of copy number variations. So what technologies can be used to do the DNA analysis in clinical research? You can use array-based approaches, you can use targeted NGS-based approaches, or you can do whole genomic or whole exomic sequencing. Then you can analyze these DNA analysis by different algorithms. You can use a combination of LOH, LST, and TAI, or you can use only LOH. Some assays use proprietary algorithms, which we don't know what the combinations are. And we can use unbalanced copy number alterations, like, for example, in our assay. And now I move to our research study. We performed an analytical validation of genomic instability metric in a series of tumor samples with known market reference, which we got from our clinical data. So we have 55 consecutive cases of tubo-ovarian high-grade serous carcinoma from the Gynecologic Oncology Center at Tuebingen University. We used DNA isolated from the same specimens that were used for market reference. We used at least 90 nanograms of DNA. The analytical validation of genomic instability metric was performed against the BRCA1/2 status, the non-BRCA mutational status, which is the other HRR genes that are coding for this variety of proteins that you saw in one of the previous slides. And we did a validation, analytical validation with the market reference with the genomic instability score. There was a limitation. 6 cases had minor material, because sometimes it's very small peritoneal biopsy samples. And in 6 cases, only 1 score was available for the genomic instability. Some of them we had at least some information on the BRCA status in the other testing modality. And usually it was because of quality of material or sample withdrawal. We're using the Oncomine Comprehensive Assay Plus as the basis. This is all known to you all. This platform can be used for a variety of complex biomarker assessments like a tumor mutational burden, microsatellite instability, LOH, or the genomic instability metric. And there's a variety of tools and targets that are being analyzed. And what we use for the genomic instability metric in the end is the copy number variations, gains or losses, and the BRCA1, 2, and HRR genes. We perform NGS on the Ion Gene Studio S5 prime on 550 chips. So genomic instability metric. Well, the previous speaker has already explained a lot of details about this technique. Just a quick recap. This is a new approach to quantifying genomic scars and genomic instability that's associated with HRD. What we perform is genome segmentation to determine copy number changes. It includes different types of unbalanced copy number events. The metric ranges from 0 to 100. The higher the value, the more genomic instability. And a GIM above the threshold for ovarian cancer, which is more or equal 16, will result in a sample being called genomic instability high. The threshold is set in the software. So here you see a typical result workup with HRD positive sample. You have here chromosomal gains depicted, and which are above the baseline, and then you have chromosomal losses where it moves down to 0 in the final calculation of the signals, and this is where we have genome loss. Here you see some of in the top some of the raw data with the signals that are then calculated into the simplified version with the bars. So it is eventually for us we are able to interpret this. Our first step was the analytical validation of the genomic instability metric in a series of tumor samples with known BRCA status. And what we find is that we have higher values of GIM in the cases with mutation in BRCA1 or 2. And what we see is we have also cases with a high level of GIM in the wild type group, which is perfectly what we expect, because we are looking, especially for these cases, that are BRCA1 or 2 wild type, or wild type of any of the other HRR genes, but still would qualify for treatment with PARP inhibitors by the status of having an HRD positive situation. Here you see some of the BRCA mutations, just for people who would want to know what are the mutations, the pathogenic mutations that you found. This is what we had in our samples. This is an example with the molecular characteristics of a sample without and with HRD. The top here is a sample without. You see that if all the signals are balanced against each other, we have almost a straight line. There's no or very few copy number variations. Here is a sample that is HRD positive, and we see several segments that are jumping up in their average line. They are jumping down, they go back and forth, and here we have a lot of signals. So this is a genomic instability metric score of 27, and this case was in concordance with a GIS score, which was at 65 here, well above the threshold of 42. If we summarize all our samples, and this is what we find a very good correlation between the data. For these are the cases that are positive for both assays. These are the cases that are negative for both assays. In the horizontal line, you have the genomic instability score, and here you have the genomic instability metrics. So what is particularly interesting for us, of course, are the cases that are in these quadrants here. So, but if we take it all together, let's look at the summary first. You find that we have 18 and 22 cases that are like both low or both high. We have 3 cases that were GIS high but GIM low. Altogether 3 cases. We have 6 cases with GIM high and GIS low, that are cases that would have been called by the GIM, but not by the GIS. So this is cases that we would have possibly allotted to therapy, and they are found in this quadrant here. When we take into consideration also the BRCA mutational status, because this is the final result, OCA plus HRD calling criteria are the following: any sample with BRCA1 mutation or GIM above or equal 16 is called HRD positive. So that's in the second table here. Similar is the approach for the reference standard. Reference standard HRD calling criteria is any sample with BRCA mutation or GIS above or equal 42 is HRD positive. So here we have this sample. So if we take all parameters together, we find that there's only 2 samples left that we might have missed with our assay, and there are 6 samples that we might have called that would not have been called with the reference standard. If we look at the ones that we didn't call, there's one that actually has a BRCA1 mutation. That is the difference between this 3 and 2. So that case would be okay. The second case is here. It's very close to the borderline area. So this is an area where we have to expect variations because we're looking at a continuous variable. So we're fine with this. This is actually the only case where we have potential issues. We are currently looking into this case some more, also looking at pre-analytical variables, and we just checked the specific samples, and there actually is a possibility. It was a very small sample, a part of the sample that was used for the OCA plus HRD testing. There was a lot of Carter artifact, and there was a lot of inflammatory infiltrate. So this is something we will definitely try to evaluate and redo this case from a different area of this patient. The problem is we have very little material. This was a patient in first recurrence, and it was basically just a small specks, peritoneal carcinomatosis with very small continuous samples. So it was taken from the same sample area, but we had a slightly different area on paraffin block with the GIS and the GIM for the testing. We analyzed that recently, and we are awaiting the final results on this more detailed analysis. The clinical research characteristics of the discrepant samples with HRD positive is, of course, we are very much interested finding out how do these cases possibly respond to PARP inhibitors, especially the ones that we would have called with GIM that were called negative on GIS. And it's a surprise to us. The cases that were GIS negative, some of them received PARP inhibitors. The reason that happened is that they received the niraparib treatment, where, as I showed you initially, also some HRD negative cases may respond. So the patient had probably a contraindication to bevacizumab, were allotted to the other treatment arm, and so finally it really didn't count what the HRD status was. So from this series, 3 patients were treated nevertheless, and these 3 patients also showed a partial response to the PARP inhibitor. We had 3 other patients that were discrepant. They received chemotherapy only. They also showed a good response to the therapy. Even response to therapy, to platinum-based chemotherapy, is a sign for an HRD positive tumor, because they're more likely to respond to this chemotherapy than other non-HRD tumors. So we take this also as a good sign, even if we don't know what the real response would have been, but because they did not receive the PARP inhibitor. Then we have a case that is in the middle, that's sort of borderline in both tests. We would recommend retesting with a different sample, for example. We have one case that was negative with us, positive in the GIS, but it had mutation, and this leaves us with that one case that we are currently working up some more regarding the pre-analytical conditions, because we are puzzled. We want to know what is the basis for this discrepancy. So what will the future yield? Here's our outlook. OCA Plus may introduce decentralized HRD testing in any lab that is using Thermo Fisher NGS, which is very useful because the Thermo Fisher NGS is a widely used platform which is already available. You don't have to establish new technology. You can use the same approaches, you can use the same pipeline. There's just a different mode of analyzing the data and using that algorithm. We are looking to harmonization studies that are ongoing to ensure comparability of this in-house assay and other in-house assays to assess HRD status. There are several assays that are established and compared to each other in the German pathology community at this point. We are awaiting a quality assurance ring study, which we don't have the results yet, that we participated in. Then we are wondering about what about other cancer types? Will the instability cutoff be different? What can we learn about this? And then, of course, we want to know what are the sources of discrepancy. We need to look at prospective studies. What is the meaning of tissue quality? What are the pre-analytical conditions? Like our sample that we had, very small and quarter artifacts, for example. That's not rarely if we find this in peritoneal biopsies. Adequate fixation, DNA quality, then tissue amount, overall cellularity and relative tumor cell content. Just like in our problem case, HRD positive cases are frequently immune hard, which means they have a lot of infiltrating lymphocytes. So a lot of the cell DNA that you find in these samples may not be tumor DNA. Then we're interested in differences in the spectrum of mutations. So are there specific mutations that lead to potential discrepancies? What do we need to look out for? And then what is the meaning of previous neoadjuvant treatment? Our problem case also was not chemo naive. This was the patient in first recurrence. So this tumor already had seen chemotherapy previously. And there are many settings where we have only very small samples, or maybe only a cytology specimen, and then the sample that we get has, from the resection, has already seen neoadjuvant treatment. We don't know how this is affecting the GIS score, and we don't know how it affects the GIM score, neither one. So what are the conclusions that we can draw from this retrospective study? OCA Plus may reliably detect HRD status in the high and low range. GIS and GIM are continuous variables. So borderline cases may need additional workup in cases of discrepancy. And third, discrepant cases with at least one test result HRD positive are most likely HRD positive as defined with response to PARP inhibitors, as far as we can say from this small series that we have looked at. And of course, we're looking forward to getting more information about the response data on the whole series of cases. We hope that we may be able to present that at some day. So we are happy with this preliminary data we have on the response at this point. So this takes me to my final slide. I want to say thank you to everybody who participated in this study, mainly Irina Bonsheim, who is my buddy in this whole journey, our molecular pathologist, and who should actually have been given this talk, but she had other obligations right now, and she will be probably talking about this topic in the future also. Then her team with Franziska Otto, Achim Rau, and Franziska Mihalik, who are all biologists working in the division, and our boss, Falco Fendt. I'm heading up the division of Gynecologic Pathology. So that's Annette Staebler, me, Robert Rotschol, who is my main co-worker, and Tanja Kopp, who is our assistant who takes care of all the specimens and the blocks who are being sent out different places, come in from outside places. She's our logistics person. Thermo Fisher, we got tremendous support from Ute Kusch, Philip Jerman, and Jose Luis Costa in talking about the results, preparing the slides, and it was fabulous to work with you guys. And we want to thank also our colleagues at the University Women's Hospital in Tuebingen, Stefan Kommos, who is the lead gynonc surgeon there, who was pushing on HRD testing on every patient that comes in, no matter what, now since this test is available. And so we have it on file for future, and he was justifying this, finding a way to justify the testing in any patient that had ovarian cancer now or in the past. Then Marcel Grube, his main co-worker, and Sarah Brucker, who is the head of the Women's Hospital. Thank you very much to everybody. It was a pleasure to give this presentation here, and I'm looking forward to your questions.
All right, excellent. And now we are back live. So I would like to welcome the previous speaker, and I would like to thank again Elaine for a presentation, as well as Professor Staebler for a presentation. At the same time, I would like to welcome as well Dr. Bonsheim. Thank you very much for joining us for these Q&A sessions. So here we have a number of questions from the audience. Let me start with the first question, which is actually for Elaine. Elaine, the audience would like to know if there's a place where they can go and have a deeper look at the Oncomine Comprehensive Plus Assay gene list, including which genes are covered by CMV, for which genes we have full SMV coverage, etc. So could you point out the audience to a place where all this information are shared, and are these information shared?
Thank you, Luca. Yes, we're happy to share that information. If you contact us through our typical channels, such as with your sales rep, we can certainly provide that information. With a panel of 500 genes, it's not very visually pleasing just to put like a list of 500 genes on the website. Therefore, it just takes up a lot of space, but we're more than happy to provide that in a format that works for you, whether it's PowerPoint or Excel, and we can certainly provide that information to you.
Thank you very much. Appreciate it a lot. Now we move on with the second questions, which could be either answered by Professor Staebler or by Dr. Irina Bonsheim, whoever likes the most. And the question is an inevitable question when we talk about HRD. Of course, a lot of people use different kind of assay as a standard or kind of reference, especially depending on which part of the words they are connecting. But specifically here, the question is about cutoff comparison between Myriad My Choice and this assay. I would like, if you don't mind, sorry, just for a second, before you answer this one, I would like to remind everybody that as of today, the Oncomine Comprehensive Plus, it's an RUO product, so it's designed for research use only. Therefore, it requires an additional validation if you want to use that as an LDT, just like the one that we have seen in this presentation. So this is an important disclaimer. Please.
Irina, you want to
So
go ahead?
who wants to take?
Yeah,
Sure.
I can say a few things. So we think that it's important still to pay attention in the borderline cases, because it's a continuous variable, and it's difficult really to make a final cutoff between cases which are positive or negative. And according to the clinical studies, each test is defining some kind of cutoff, but we think that it's still there are the cases which are in the borderline area. There's still not clear knowledge how to perform with those cases, and we also had a lot of discussions now in the German ring trail how to really pay attention, or what to say to those cases which are in the borderline area. So maybe Annette, you also want to say something? Or
Yeah, I think for the time being, we just have to pay attention to the individual cutoffs that have been defined as the statistical best possible cutoff for each test, which for the OCA Plus is the score of 16 or above. And I would be reluctant to call a case that is a score of 15 or 14, maybe 13, to call that outright negative. Honestly, if I was really eager about giving this patient an option, I would try to repeat this test on another sample, another location from this patient, for example. Secondly, I can add the case that we had the major discrepancy at the time of recording of this talk, which is now already almost 3 weeks ago. Irina and her lab has retested that case from another sample, and we now have a score of 10. So pure sampling was not the issue, but now we need to look into the different the genes that are potentially affected. And then the third thing, Irina, I haven't mentioned, but I have to congratulate her and her lab. We have actually passed successfully that German quality assurance ring study. So we are basically, in some sense, officially certified or approved to perform the test now. And alongside with that in-house validation and with the knowledge that we pay very close attention to the cases close to borderline, below or above borderline, I think we can confidently perform this assay now in our day-to-day routine.
Thank you very much. Actually, this was another question that just came in. Many people said during your presentation you were still waiting for your result. It seems like you have received them now, and they seems to be very positive, which is yet another important confirmation that reassures about the validity of using the GIM score. Of course, I think the entire EMEA region was looking and waiting to the German ring trial. And the question is, do you know whether these data will be disseminated publicly? And do you have an idea about the timeline around that?
I have no idea about the timeline, but a lot of results from the ring trials are published. So I suppose that this, because it's an interesting topic to everybody in the community, it will be published soon.
Okay, thank you very much. So we'll have to stay tuned, so to say, a bit more in order to get a better picture. But again, it's reassuring to know that you have passed the test, and congratulations, of course, about that. Now there's one last question, as we are almost at time. I would expect, again, this is a very tricky and typical question. But, you know, as we have here no filter, and we always would like to make the audience happy, that's a typical question when it comes to ion torrent, and it's about homopolymeric region. It's something that it's kind of following us forever and ever. And the question is that, did you have any problems with that while picking for your BRCA1 and 2 mutation, or have you found any kind of difficulties or limitation during your HRD testing?
So I'm doing Thermo Fisher sequencing for many years now, and I have to say that the problem with the homopolymers is changed. It got better with changing chemistry, things like that. But of course, there are still cases which are difficult to evaluate, as the mutations in the homopolymeric region can be true biological or biologically, but they can also be an artifact. And in those cases, we always look at the raw data to see if to see how high the frequency is, and mainly we get a good decision, I think, but there with low tumor cell content, there may be still a very low percentage of cases where it's hard to decide if it's a technical artifact or it's really a mutation.
All right. Well, thank you again for your transparency in here. I guess passing the Ring trial is once again an reassurance that probably you are well on this problem and able to control that. But once again, I would like to thank the speakers and the participants to this Q&A session.